let's do a final accounting of how much energy or how much ATP is produced during cell respiration. You'll see on our last video about aerobic cell respiration that we estimate how much ATP we make during oxidative phosphorylation. We estimate at 32 to 34 ATP. But why is it impossible to get a straight answer about how much ATP is produced per glucose molecule? Why can't I say exactly how much ATP we're making per glucose molecule? And that's due to the decoupling of glucose oxidation and oxidative phosphorylation. So as we're losing our electrons from glucose, as we're losing those hydrogens, we're not directly making ATP. We're passing those hydrogens on to our electron carriers. So we're not breaking down glucose and immediately making energy. We have different steps in that pathway. And because of those steps, we don't have an exact number of ATP we're making. So we make roughly about 3 ATP per NADH. So for each NADH, we make about 3 ATP. And then for each FADH2, we make about 2 ATP because FADH2 enters a little bit lower down in the electron transport chain. Let's look at our efficiency. So anaerobic metabolism is less efficient than aerobic metabolism. If we consider a hypothetical maximum of 36 ATP, because we're going to round it up just a little bit, because remember we're about 32 to 34. So let's say that we have a hypothetical maximum of about 38 ATP that are produced when we do aerobic cell respiration. Aerobic cell respiration would be 19 times more efficient than fermentation, because fermentation through glycolysis, or glycolysis coupled with fermentation, only makes 2 ATP. So 19 times 2 would be our 38. So oxidative phosphorylation through aerobic cell respiration is much more efficient than just using glycolysis. Also, let's look at aerobic cell respiration in depth in terms of its efficiency. Aerobic cell respiration is about 40% efficient in terms of converting chemical energy into ATP. So we get about 40% of the energy that we could get out of it, possibly. But your car is only 15% efficient. So you might think that 40% is not so great, but it's a lot better than your car is. Your car is only 15% efficient at converting the chemical energy in gasoline into mechanical energy to run your car. Now let's look at other things we can break down besides glucose and how we can regulate metabolism. So cell respiration is very versatile. We focus on glucose during our past discussions, but it turns out all macromolecules can be used as substrates for respiration. So you don't only eat sugar in your diet, you eat other things. You eat protein, you eat hamburgers, you eat vegetables, carbohydrates, and whatnot. So all macromolecules can be broken down in cell respiration. But different components will enter the process at different points. So different components or different things you eat, different molecules will enter our process of cell respiration at different points. Now, this is just an interesting fact. If an animal is starving, this is the order of digestion of stored molecules. So this is how your body breaks them down. For about 3 days, your body can live off of carbs, okay, just eating carbs stored in your body or glycogen. Then for about 3 weeks, they can survive on stored fat reservoir. So if you have any fat storage on your body. But then after that, after about 3 weeks and beyond, you're about to die, and your body will begin to break down the proteins. It'll break down your muscles and other organs. Now, let's look at regulation. Because there are so many enzymes that help mediate respiration, since every step of respiration is mediated by an enzyme, there are multiple opportunities for feedback to control the process. So because we have so many enzymes that mediate different parts of cell respiration, if we change how those enzymes are working, then we can promote or inhibit cell respiration. For example, we have the enzyme Phosphofructokinase. Remember, ACE, the suffix ACE is a sign that we're talking about an enzyme. So Phosphofructokinase is a glycolysis enzyme. So it's an enzyme involved when we're originally breaking down our glucose and glycolysis. Now it's one control point. Okay, it is stimulated by AMP and inhibited by ATP and citrate. So when we have phosphofructokinase and we have a lot of AMP, and remember, AMP is low energy. So when we're low on energy, okay, because we don't have ADP or ATP, phosphofructokinase is stimulated. So that's going to stimulate glycolysis. But when we have a lot of ATP and citrate, meaning we have a lot of energy and we have citrate to go through our citric acid cycle, we don't need to be breaking down more sugar if we already have a lot of these materials. So that inhibits phosphofructokinase. And now that's it for our discussion on cell respiration and chemoheterotrophic nutrition.